figure that can give you an idea of which fuel sources are used during exercise and how long they last. So here we have a chart. We've got metabolic fuel on the y-axis, and on the x we've got our time in both seconds, minutes, and then hours. So let's start running a marathon. In that first few seconds of exercise, we're going to use some stored ATP, creatine phosphate, and aerobic metabolism to provide our energy. After a few seconds, however, the stored ATP becomes depleted, and anaerobic metabolism and also maybe some aerobic metabolism will take over. Once the body realizes that the exercise will continue, the body's going to ramp up aerobic metabolism, so the green line here. Now, when exercising for hours, such as in a marathon, both glycogenolysis, or the breakdown of glycogen, and free fatty acid oxidation are going to be used to make the ATP in aerobic metabolism. Now, stored glucose is conserved until you readily need it, such as when you're sprinting for the finish line. Now, I do want to say that this graph would be what you would expect when running a marathon. It would look totally different for weightlifting, for example, than it would for running a 1 kilometer sprint or a 27-mile marathon, or like cycling race that has steep hills, causing you to alternate between aerobic and anaerobic metabolism. So this is assuming you're at a nice constant pace of aerobic metabolism, like when running a marathon, for example. So this would be an example of a metabolic fuel use. So how does the body react when fasting or starving? Now, in these scenarios, the priorities are to supply enough glucose to the brain and the red blood cells so that the body can maintain its basic functions, and also to preserve protein used to prevent muscle breakdown. So let's create a scenario. You decide to binge on fast food cheeseburgers. After your third double cheeseburger with large fry, you step on the scale. Now you realize that you weigh way too much. You now decide that you want to not eat for 5 days to lose some weight. Now in the fed state, immediately after eating those burgers, insulin is going to be used to stimulate the storage of lipids, proteins, and glucose into cells. Now, assuming plenty of oxygen is present, both glycolysis and aerobic respiration will occur. So notice here that your energy source is going to be glycolysis and aerobic respiration. However, in the fasting state, glucagon is going to be produced. Remember, glucagon is released when you're fasting, low blood sugar levels. Now, what does glucagon do? It's going to cause the body to use fuel reserves to maintain its bodily functions. Specifically, hepatic glycogenolysis is the main way the body refuels after fasting between meals. Now, this is supplemented by gluconeogenesis and fatty acid metabolism. However, unfortunately, glycogen stores only last about a day, so they may be effective, yet not for the long term. So after that first day of fasting, you're now going to enter the starvation phase, in which you must use other sources of fuel to make ATP. Now, like we saw before, glucose levels are going to be maintained by hepatic glycogenolysis. After most of the hepatic glycogen has been consumed, the body must now get creative and find alternative sources of energy. Adipose tissue start releasing free fatty acids for beta oxidation. This means that muscle and liver now switch their fuel source from glucose to now fatty acids. The liver can also use these fatty acids, as well as lactate and alanine, for gluconeogenesis, which is especially useful for the red blood cells since they can't use the fatty acids or the ketones. Now, why can't these red blood cells use the fatty acids or ketones? It's because they lack a mitochondria, so they need glucose instead. They're kind of the purists of the body. Now, after day 3 of starvation, the ketone bodies become the main source of energy for the brain and the heart. During this phase, about 2/3 of the energy will come from the ketones, while about 1/3 is from glucose. Now, since the ketone bodies are made from fatty acids, the amount of stored adipose tissue determines your survival time, assuming you have access to water and, other important minerals and such. Once adipose stores are going to be depleted, vital proteins are degraded to make energy, which doesn't take long to result in end organ failure and then thus death. Now, what's the moral of the story behind all this? Don't binge purge on fast food because starvation makes your body scramble for different sources of energy. In the last part of biochemistry, we're going to talk about cholesterol metabolism, lipid transport, the lipoproteins, and the dyslipidemias. Now, before you get too mad at me, we've already seen this pathway. This is nothing new for you. When we talked about this pathway earlier, we were talking about ketone bodies. I said that the enzyme HMG-CoA synthase was an important enzyme for two reasons. It's used in the creation of ketone bodies, which we already covered, but it's also going to be seen again in cholesterol synthesis. Well, the title of the slide is cholesterol synthesis, so here we are. Remember that there were two forms of the enzyme HMG-CoA synthase. The mitochondrial form acts on the ketone bodies, and the cytosolic form is going to be used for the synthesis of cholesterol. So, let's clear this off, and let's talk about cholesterol. Cholesterol is going to be an important part of cell membranes, and it's also used as a precursor to the bile acids, steroid hormones, and some vitamins. It can be synthesized in the smooth endoplasmic reticulum of any cell in the body, but primarily it's made in the liver. Once cholesterol has been synthesized, about 2/3 of the cholesterol will be esterified by the enzyme Lethicine-cholesterol acyl transferase, or LCAT for short. So what's the point of this esterification by the LCAT enzyme? Well, esterification of cholesterol allows it to be stored inside vesicles rather than being embedded in the membrane, which is important so that it can be transported through the blood to other parts of the body. Here is going to be a big, complicated, and truthfully useless chart for cholesterol synthesis. Let's simplify it down further here. As I mentioned earlier, acetyl-CoA is used to make HMG-CoA, which can then be used to synthesize either ketone bodies or cholesterol. So here's our basic chart. In fact, HMG-CoA reductase here, our enzyme, is going to convert HMG-CoA into mevalonate, which is the rate-limiting step in cholesterol synthesis. And therefore, it's going to be a great target to regulate cholesterol production. So if we cross out this enzyme, we're going to use the statin drugs. These are the drugs that have the suffix statin. Now, statin drugs such as lovastatin competitively and reversibly inhibit this rate-limiting enzyme HMG-CoA reductase. So if you inhibit this enzyme, you decrease cholesterol synthesis by decreasing how cholesterol is made. Now, the boards love this enzyme HMG-CoA reductase, so make sure you commit this one to memory. Now let's spend some time with the tougher concept, lipid transport. We'll explore this graph in more detail in just a bit, but for now I'm going to show you the big picture. And we can also cover some basic concepts. Because fat and cholesterol are hydrophobic, meaning they don't dissolve in the blood, lipids require carrier molecules to enter and move throughout the circulation here, just like you see here. Fat from the diet and from the liver associate with amphiphilic proteins called apolipoproteins, which act as structural proteins and surface markers. So let's take a look at this math equation. Together with other lipids, including cholesterol, cholesterol esters, triglycerides, and phospholipids, the apolipoproteins form lipoproteins. So let's do a quick little math equation. Apolipoproteins plus these lipids equal lipoproteins. Now, what are the different lipoproteins that we'll need to cover? These lipoproteins will include our VLDLs, our IDLs, LDLs, and HDLs. So there are several key enzymes in lipid transport. Pancreatic lipase degrades dietary triglycerides in the small intestine. Specifically, it cleaves triglycerides at positions 1 and 3, giving rise to free fatty acids and monoacylglycerol. Here we see a triglyceride that meets pancreatic lipase, which degrades it into long hydrocarbon tails, which are free fatty acids. These can then enter enterocytes and reesterify into a triglyceride that can then be transported in a chylomicron. We've got another enzyme here. It's going to be lipoprotein lipase, and this lipoprotein lipase is on the vascular endothelium of adipose and muscle, and it's going to catalyze the same reaction as pancreatic lipase in the gut. It's going to degrade triglycerides that are circulating in chylomicrons and VLDL so that they can be absorbed by local cells, and the chylomicron remnants that are going to be transported back to the liver. Hepatic triglyceride lipase, or HL, degrades the triglycerides in IDL to form LDL. Again, the enzyme takes a triglyceride, so notice how we're going from an IDL to an LDL. The enzyme is going to take a triglyceride and cleave it into free fatty acids. Alright, and lastly, hormone-sensitive lipase is an intracellular enzyme, which is going to degrade triglycerides, mostly in adipocytes. Do you remember what the function of LCAT was with regards to cholesterol? As I mentioned a moment ago, LCAT, or lecithin-cholesterol acyl transferase, is going to esterify cholesterol. So here's our reaction that we saw earlier. It's going to esterify cholesterol, which allows the cholesterol to enter the core of the lipoprotein, where it can get stored for transport. About 2/3 of cholesterol in the circulation is going to be in this esterified form. Now, this enzyme is also required to get cholesterol into HDL to make a mature HDL molecule. HDL that is not loaded with cholesterol travels from the liver. It's going to meet LCAT and load up on cholesterol. Here we see the loaded HDL molecule, and it can now take two paths. It can go back to the liver, effectively reducing the amount of cholesterol systemically, or it can transfer cholesterol esters to other lipoproteins. CETP, or cholesterol ester transfer protein, can then mediate the transfer of cholesterol esters to other lipoproteins such as VLDL, IDL, or LDL.